because we're mitochondriacs at MIHRA, we are going to talk about now more mitochondria with our esteemed colleague, Dr. Christian Lood. Thank you, Christian.
Thank you, Leslie. I'll continue on the same topic as previous speaker here and discuss how our power plants, the mitochondria, may be on the go in myositis. Next one, please. So as noted in prior talk, mitochondria, we usually think about them as these power sources, and they're inside of cells producing a lot of energy, which is important for our cells, not least our muscle cells. But however, it's worthwhile noting that we don't think that mitochondria are human. We think they come from bacteria originally. They coexist with us. And as such, they also contain many things that look like bacteria. And since they look like bacteria, our immune system is very intrigued by these mitochondria. However, the crux here is that unless we think about the inflammasome that Dr. Naddaf mentioned, our immune system will not see mitochondria if they are still within cells. But if they are seen by the immune system, they can evoke autoimmunity. We see a lot of autoantibodies towards mitochondria in different myopathies. And we know that these mitochondria can promote inflammation. But would it be that a mitochondria would be seen outside of a cell? Are they on the go or do they stay inside of cells? Next, please, Leslie. So we have reason to believe, both in myopathies and also in other inflammatory diseases, that mitochondria do not stay within cells, but they actually move. They move around. And we can see this in inclusion body myositis, where we see in the fibers that they are COX negative, meaning they lack mitochondria. Where are the mitochondria going? We see in JDM in the lower left page, using electron microscopy, that muscle tissue are pumping out, throwing out mitochondria into the extracellular tissue, depositing them for our immune system to see them. And we have published a paper describing on why and how that is happening. But also in our peripheral blood, our immune cells, like neutrophils or platelets, they can also, when activated, release these power plants that are now inflammatory, looking like a bacteria in your blood. And one of those important molecules that they release, we refer to as FMET here in red circles. So this is, of course, all theoretical. Do we then have evidence that this is occurring in patients, and what are the implications of that happening? So next slide. So we have measured these mitochondrial peptides in the blood from a ton of different patients and different disorders, and we do find in both juvenile dermatomyositis, adult dermatomyositis, and inclusion body myositis that patients have elevated levels of these mitochondrial markers in blood. So it's something we can measure in the lab. And this seemed to reflect very well muscle function. Though we may not agree that MMT8 is a good marker or not, this is a soluble marker you can measure in blood that even in IBM seems to correspond to muscle function. And this would be one of the first, if not only, good soluble marker that we have for IBM to study disease activity or muscle function. So thinking now about us having all of these mitochondrial markers, mitochondrial components, if you would, in blood, the question is, do these contribute now to peripheral systemic inflammation in these patients? So next slide, please. So this is going to be a little bit messy, but I'll try to lead you through it just to give you an overview of what we're trying to accomplish with this project. We do try to bring novel biomarkers to the market to try to help investigators and clinicians to monitor better disease activity and so forth. But when it comes to long term, we try to identify new mechanisms of how inflammation and damage occur in patients, trying to develop new targets to treat. And we do know that these mitochondrial peptides, FMET, activate immune cells through a receptor called FPR1. There's a drug we can use called cyclosporin H, which blocks this specific pathway. Oh, sorry, Leslie. And what you see to the right is that we have found that serum or blood, if you would, from patients with inclusion body myositis and dermatomyositis are inflammatory. They evoke an immune response. But if we add in that system cyclosporin H, so this mitochondrial peptide inhibitor, to that system, we normalize it. We remove all of that inflammation from the peripheral blood. So we do believe that blocking this pathway, which we have shown in other diseases as well, could be helpful and a new target to consider in myopathies. So thank you, Leslie. Next, the final one. And this is just to summarize and saying that we believe that mitochondria are not only inside of cells, but they are on the go in myopathies. And we can use these mitochondrial markers in blood and tissue to help monitor disease activity, assess prognosis, and guide treatment. And we believe that targeting these mitochondria and making sure that they stay where they're supposed to do may alleviate inflammation in myopathies. And then finally, on the last slide, I would like to just acknowledge all of my team members and collaborators for all these projects, funding, and not least MIHRA and TMA for all of their support on this. Thank you.